Namaste viewers. Today I'm before you to present first semester BSc Biotechnology Cell Division Meiosis. I request you all to subscribe this channel. Now let us go to the introduction part. Meiosis is a division which occurs in diploid cell and gives rise to four haploid cells or nuclei, each having half the number of chromosomes as compared to the parent cell. Okay, meiosis, also known as reductional division. This term meiosis for the first time introduced by Farmer and Moore in the year 1905. The process of meiosis was studied by Strasburger and Sultan. Meiosis, so in this case, meiotic division can be seen in two successive stages, okay, namely meiosis first and meiosis second. Each of these show karyokinesis and cytokinesis. These two steps involve following stages, okay. Hope you're making note of all this information in your notebook. I request you to invest your quality of time, okay, while watching video. Meiosis can be differentiated into meiosis first and meiosis second. So in these two stages, you are going to study the meiosis. Okay, in both the case, you can see the formation of karyokinesis and cytokinesis. Okay, here also karyokinesis and cytokinesis. Whereas in meiosis first, you can see the prophase first, metaphase first, anaphase first, and telophase first. In meiosis second also, you could see prophase first, second, metaphase second, anaphase second, and telophase second. Whereas in this prophase first, you are going to observe the further substages like lactotine, zygotine, pacatine, diplotine, and diakinesis. Now let us go through the prophase first. Okay, this is studied under five substages. Leptotene, in Greek that is leptos means slender, tene means that is thread. During this stage, the chromosomes are thin, long, and uncoiled. Each is a double chromosomes consist of two chromatids. These chromatids are held firmly. Each chromosomes appear as a string of beads. The beads are chromosomes. Okay, you can see here this particular leptotene microscopic observation can be seen here. Okay, here the chromosomes first become visible. Okay, and later you can see the leptotene like this. Now let us go to the second stage, that is zygotine. Zygotine in Greek, zygo means male, tene means thread. During this stage, the homologous chromosomes pair, that is half are maternal and half are paternal. This pairing called as synapses. Okay. The pairs are called as bivalents. You can see the pairs, right? These are called bivalents. Each bivalent consists of four chromatids and therefore called as tetrad. Okay, two chromatids of some same chromosomes are known as sister chromatids. Two chromatids of different chromosomes are non-sister chromatids. Okay, synapses are homologous chromosomes pairing begins in this particular stage of zygotine. Now let us go through the pacatine. In Greek, pacus means thick, chain means thread. Here the pairing is complete. The pair chromosomes are bivalent, becomes short and thick. Okay, coiling takes place between non-sister chromatids of tetrad and segments are exchanged here. Okay, this process is called as genetic crossing over. It results in the recombination of genes. Crossing over takes place by breakage and reunion of chromatid segments. Breakage of chromatid segment assisted by an enzyme that is endonuclease, and reunion helped by enzyme or protein. The reunion is called as annealing. After crossing over, the sister chromatid of chromosome become dissimilar. The points of interchange are X-shaped and are called chiasmata. Okay, this crossing over you're going to observe in the form like a chiasmata. Okay, you can see here this is the microscopic view of pacatine. Now let us go through here diplotene. In Greek, diplo means double, tene means thread. Here, chromosomes show high degree of condensation. Repulsion between homologous chromosomes begin in this particular stage. Each tetrad now appears in different shape. Repulsion results in thermalization of chiasmata. Okay, and finally they disappear, and you are going to observe the diplotene structure of microscopic stage will be like this. Now, the next one is diakinesis. Greek dia means across, kinesis means movement. Difference between diplotin and diakinesis are not very clear. Here, chromosomes show the degree of condensation and fewer chiasmata seen than diplotin. This is because of terminalization process can be seen here. At the end of diakinesis, nucleolus break down and disappear, then spindle fibers appear, nuclear membrane breaks down and chromosomes are released into the cytoplasm. Okay, and the next you'll be able to see here the different stages of meiosis, meiotic prophase. Till now we went through the leptothene, right? Chromosomes are somewhat that's called bead chromosomes. Okay, then zygote, that is bivalent, pachythene, pachynyma, tetrad, then diplonyma, you can see the chiasmata, and diakinesis, you can see the terminalization. Okay, this can also be mentioned as in the meiotic prophase first leptonyma, zygonyma, paranyma, diplonyma, and diakinesis. Okay, hope you are making note of all this. Okay, if you are unable to go through this video within time, you can pause this video and you can make a note. Okay, now let us go through the crossing over. During prophase first of meiosis, part of chromatid of one chromosome and vice versa, this interchange of the parts of the chromosomes of a pair of homologous chromosomes known as crossing over. Crossing over is a process by which two chromosomes of homologous pairs exchange equal segments with each other. Okay, you can see the equal segment they have exchanged with each other. Crossing over occurs in the first division of meiosis. Okay, now let us go through in detail here about the synapses pairing of homologous chromosomes. Okay, here you can see this one is paternal and other is the maternal. Okay, homologous chromosomes in a tetrad cross over each other. Okay, here in the second step, pieces of chromosomes or genes are exchanged. Okay, there are pieces which are exchanged each other, and this is the crossing over, right? Or chiasmata. And this produces genetic recombination in the offspring, and you'll be able to see the variations, right? Which gives the new genes, right? Or new combinations can be seen over there, right? Both the side you can see that particular crossing over, right? Crossing due to the crossing over, you can see the recombination. Importance of crossing over. Crossing over takes place between the non-sister chromatids of homologous chromosomes. Okay, sister chromatids means that's the same chromosomal arm as non-sister chromatids means other chromosomal arms. Crossing over is one of the most important events in meiosis because it allows variation in the produced, which is responsible for the offspring. Variation in the produced offspring can be observed. This produces new individuals having new combinations of traits and helped in establishing the concept of linear arrangement of genes. This crossing over also helps in the mapping of chromosomes and then selection of useful recombination. So this is all about importance of crossing over. Okay. Now let us go through here metaphase first. So metaphase first is the second phase of meiosis, right? So the tetrads from prophase first line up in the middle of the dividing cell randomly. Spindle fibers attach to the tetrads from both the ends of the cell. Right? You can see this is the metaphase plate, and here you can see the meiotic spindles can be seen. Then anaphase first. Anaphase first begins when the two chromosomes of each bivalent separate and start moving towards opposite poles. In anaphase first, the sister chromatids remain attached at their centromeres and move together towards the pole. Okay, and here you can see the chromosomes homologous separate. Telophase first. The homologous chromosomes pairs reach the poles of the cell, right? The homologous chromosome pairs complete their migration to the two different opposite poles. A nuclear envelope reforms around each chromosome set. The spindle disappears and cytokines follows. Okay, so this you can see the diagrammatic representation here. Then coming to the figure of meiosis. Okay, this is the interface of meiosis chromosome duplicates here. Then prophase first, Okay, in prophase first, you'll be able to see almost all the sub stages can be seen over there, right? Like chiasmata, diakinesis, and sister pairing chromatids, everything you'll be able to see. See, you can see here the chiasmata, right? Exchange of the sister chromatids can be seen here. So whereas here homologous chromosome that is red and blue pair and the exchange segment that is 2n is equal to 6 it can be seen. This will be tetrad. Okay, and next here metaphase first centromere with kinetochore all the chromosomes which are aligned in the equatorial plate of this particular cell, right? And microtubules attached to the kinetochore that helps responsible for the movement of the chromosomes to the opposite poles during the anaphase stage, right? You can see the metaphase plate. Whereas in the anaphase first, homologous chromosomes separate each other, okay? After the metaphase, then pair of homologous chromosomes split up and move apart. You can see sister chromatids remain attached to the particular spindle fibers. Here you can see the centrioles pairs, then homologous chromosomes crossing over can be seen here, right? This is in the prophase first, and one pair of homologous chromosomes, right, which will separate. Here you can see the metaphase first, and one set of the chromosomes, and here homologous set of chromosomes. This is anaphase first. This anaphase first thus gives rise to a telophase first, right? And further, in the phases of meiosis second, mitotic division of two haploid cells to produce four haploid daughter cells. Okay, here you can see the different stages like prophase 2, metaphase 2, anaphase 2, and telophase 2. You can see the prophase 2, metaphase 2, then anaphase, telophase, and tetrad of microspores can be seen. Meiosis second begins without any further replication of the chromosomes. Okay, in the prophase second, the nuclear envelope breaks down and the spindle apparatus forms. While chromosome duplication took place prior to meiosis, okay, of meiosis first. No new chromosome replication occurs. Before meiosis second, the centrioles duplicate, okay. This occurs by separation of the two members of the pair and then the formation of daughter centriole perpendicular to each original centriole. The two pairs of centrioles separate into two centrosomes. The nuclear envelope breaks down and the spindle apparatus forms. Now let us go through metaphase 2. The chromosomes become arranged on the metaphase plate. Okay, centromeres are arranged in a line called equatorial plate of invisible spindle apparatus. Previously occurring nuclear membrane is not present here. The centromeres separate and the sister chromatids now individual chromosomes move towards the opposite poles of the cell. The separate chromatids are now called chromosomes in their particular own condition, right? Okay, meiosis second can be seen here, and that's in the anaphase 2, separation of sister chromatids and move to the opposite poles. Here you can see the microscopic view of the meiosis anaphase 2. Nuclear envelope forms around each set of chromosomes. Okay, nucleolus disappears. Nucleolus appears in each nucleus. Chromosomes lengthen and become indistinct. Okay, here you can see the diagrammatic representation of telophase 2. So here you can see the meiosis 2 here. Okay, this meiosis 2, which is in turn responsible to give the daughter cells. Different stages in meiosis first and meiosis 2 here. This is early prophase first, okay, then middle prophase first, okay. Then here is the same thing again. Spindle formation can be seen again the middle prophase first, late prophase first, metaphase first, then anaphase first, then telophase, okay, then telophase first to prophase 2, then metaphase 2 here, then here anaphase 2, telophase 2, and this is the product of meiosis, which are in tetrad condition. Kindly make this a pencil sketch in your notebook. Okay. Now let us go to the importance of meiosis. This produces haploid gametes so that the diploid number of the species remains constant generation after generation. Source of genetic variation because crossing over brings together new gene combinations on chromosomes. So these are some of the importance of meiosis. Hope you are able to make a note in your observation book. Now let us go through the differences between the meiosis and mitosis. Mitosis occurs in body cells, whereas meiosis occurs in the reproductive cells. Number of chromosomes remains the same in the daughter cells. Number of chromosomes is held in the daughter cells. Daughter cells are identical to parent cells and each other. Daughter cells are genetically different to parent cells and each other. Here in mitosis, two daughter cells are formed, whereas in meiosis, four daughter cells are formed. Okay, in mitosis, homologous chromosomes do not come together. Okay, whereas in meiosis, homologous chromosomes come together. There is no exchange of genetic material between the chromosomes, whereas in meiosis, there is an exchange of genetic material between the chromosomes. Okay, this is all about the differences between mitosis and meiosis. Hope you understood today's concept about meiosis. I, Dr. Pratibha K, signing off for today. Stay tuned with me and stay connected. Stay safe. Thank you for watching this video.